you, Jose. That was an outstanding talk, an outstanding introduction to the F and the G proteins that are so important for initiating infection. Yeah, thanks. So here's our little cartoon of the same. Both of those proteins are on the surface of the virus, and the G starts everything by binding to a receptor. We've assumed all along, and then the F protein does this fusion magic. So I want to show you this picture. I came into the lab one day in graduate school. Seymour Levine was my mentor, and he showed me this, and he says, "Look at this. I've never seen anything like this. This is a tracing of purified virions, RSV virions, that were radio-labeled with either protein, which is the solid line, or sugars, which is the dotted line." And he said, "Look at this one protein. It's got a huge amount of sugar on it." So this turned out to be the fusion protein. You can see how much protein is there and how much sugar, a lot more protein than sugar in this relative experiment. And it's just the opposite here with the G protein. Just a huge amount of sugar. It's about 60% sugar, we know now. And if you look at it in a regular slab gel, you can see the same thing. A little bit of protein and a lot of sugar compared to the F with a lot of protein and a little bit of sugar. And if these virions are treated with a protease, both of these are lost. Both of the fusion protein and the G are lost. Now, we didn't know we just knew they were on the surface at this point. We didn't know, and this is now going back even further than Jose. Sorry to say, but this is like 40 years ago. And I don't want to mean it's embarrassing. This was a fun time. This was exciting to find these new things. This is now what we would imagine the protein to look like with so many sugars. Our A2 strain that we study in our lab, the yellow is the unlinked sugars. There are 4 of them. But all this gray is the O-linked sugars. We estimate probably about 35 O-linked sugars. And those are put on serines and threonines, not all of them, but quite a few of them. So a lot of the work early on, of course, was done in tumor cells, HeLa cells and Hep2 cells. But in fact, RSV will infect, in my hands, most every cell in culture. It's very promiscuous, which kind of doesn't make sense for a very specific pathogen for the human respiratory tract. Turns out that the receptor here on these cells is heparan sulfate. It's a repeating disaccharide that's present on most cells, and so cells in culture, so immortalized cells at least. So it's everywhere, and that's probably why the virus can infect these cells. We think of that now as a surrogate cellular receptor. So that's what it can use in these cells. That's why we could see the infection, but we don't think that's the real receptor in vivo. Of course, the F protein in the cells, these are individual cells. When a cell's infected, it begins fusing with its neighbors, and you get these giant syncytia, which are the middle name of the virus. When I did a sabbatical with Peter Collins 20 years ago, he was gracious enough to let me put the GFP gene into this virus, which we use constantly, and I know probably many of you in the audience do. It's a really useful virus to be able to follow virus infection in real time. But really, to know what's going on in vivo, we needed a system, a culture system that would allow us to look at something similar to the in vivo situation. And these are cells where we began working, Peter and I, with Ray Pickles at University of North Carolina, where they grow these cells in the air-liquid interface. They appear and function in every way we can measure as the real in vivo airway epithelium in the trachea, the bronchi, and the bronchioles where RSV infects. So they have ciliated cells, and you can see sort of the cilia up here. They're also producing mucus, and mucus covers these cells. It takes quite a while. They also have tight junctions, actually, between the cells, so they're very tightly associated. It takes quite a while to differentiate these in culture, but it can be done, and it turns out that it's worth doing that. One of the first experiments that we did was to inoculate these cells with the virus apically. If we did it basolaterally, we didn't see any green cells. But if we do it apically, you can see the green cells here. And the cells that are ciliated have this red staining for the cilia. So it infects the ciliated cells, the apical cells. The other thing that was interesting is we don't see syncytia fusing these cells together, and it's probably because of the tight junctions preventing the F protein that goes to the apical surface from going to the neighboring cell. Now, when we look for heparin sulfate in these cells, you don't see it on the apical surface. It's not detectable. It is detectable on the basolateral surface. So it doesn't seem then that heparin sulfate is likely to be the receptor in these cells, since it's not on the right cells. So just to prove this, or to confirm this, we can mix heparin sulfate with the virus and then put it on cells. When we do that on HeLa cells, it blocks infection by about tenfold at this concentration. If we do the same thing on these human airway epithelial cells, it does not. And so again, that suggests that this is a different receptor. The heparin sulfate would actually be binding to G and preventing it from binding to the heparin sulfate on the cells here, but not here, probably because heparin sulfate's not the receptor there. So it confirms that there's another receptor on these HAE cells. Now, the next work that I'll tell you about is from Sarah Johnson, who was a grad student that finished last December. So she had actually done this experiment and the next several that I will show you. There is a monoclonal antibody, very famous, which binds to the G protein 131.2G that Larry Anderson made years ago. It has been described as a non-neutralizing antibody. It doesn't neutralize in any of the cell cultures that we traditionally use, but what about in these HAE cells? So when we do the same experiment, putting 131.2G in, again, it does not neutralize in HeLa, but it does neutralize in these human airway epithelial cells. So this antibody is blocking infection. We think G does primarily attachment, so it's likely that this is blocking the attachment. So if you look at those two experiments, this one and the one before, 131.2G inhibits infection of HAE, not HeLa. Soluble heparin sulfate is the opposite. So they're really blocking two different things. So we wanted to test G-neutralizing antibodies, another one. So this one was from Ed Walsh. It's called L9. It was actually isolated against the long strain. We use it here against A2. And it does neutralize in HeLa cells. So we do the same experiment. We do the HeLa cell infection, and it does neutralize by about tenfold. When we do the same thing on HAE cells, it neutralizes 100-fold. So it's much more potent in these HAE cells, these primary cells. I'll come back to that in a minute. But this is consistent, again, with different receptors, and also that the heparin sulfate binding site and the site that binds on HAE cells is close together, because this one antibody will block both. This is our sort of working model of the protein. It's not really drawn to scale, but it's the parts that we're most interested in are up here. These are the parts here that have so much sugar on them, and we won't talk about those. They make the protein look like a mucin. So many sugars there. So the G sequences are highly variable, as Jose has alluded to, We all know the two large mucin-like domains. Now it attaches to heparin sulfate via this heparin binding domain over here, and Judy Beeler showed that years ago. This is a highly basic amino acid region, so it has the opposite charge of heparin sulfate, which is negatively charged. This is a positively charged region, so that makes sense. So we have then the conserved motifs up here at the top. There's a region here that's completely conserved among all G proteins. There's a cysteine loop structure here, which was solved by Jeff Gorman, where these 4 cysteines bind in this pattern. And then the CX3C motif here, 2 cysteines with 3 amino acids in between, which Ralph Tripp recognized as being a chemokine binding, well, at least a chemokine indicating sequence. There's only one chemokine like it, and it's called fractalkine, and it binds to a receptor, but it's got this CX3C motif. So we want to look at where monoclonal antibodies, these 2 monoclonal antibodies bind. Ed Walsh, when he made L9, had isolated mutants that were resistant to it. And so here's the wild-type virus. These are purified virions, the wild-type virus, and then these 2 mutants. You can see that all of them have some G protein in them. Now, what about staining with the L9 antibody? We expected we would not see it because these had been selected against L9, so the L9 does not bind. Surprising thing was that 131.2G didn't bind either, suggesting now that these 2 antibodies are binding in a similar region. And if you look here at the amino acids that have changed, there are actually 4 changes in each of these mutants, but 2 of them are in common, F170L or F170 and C186. So these are 2 amino acids right here in the neck. So it seems like one or the other of these is important for binding of both antibodies. And in fact, what we did was to do the same kind of experiment, but using a protein that has a mutation in one or the other. And in fact, just like this picture looks, in both cases, neither antibody bound. So it looks like both antibodies are binding right in the same region. We don't think they're binding in an identical site because one neutralizes in HeLa cells and the other one doesn't. But still, they're overlapping probably in this region. This is as far as we've gone so far here. Now, what about a disruption in this region? This was a virus that Mike Teng made, which you can see will, whereas wild-type virus, wild-type virus can infect both cell types equally well, but this C186 infects HeLa fine, but not very well HAE cells. So this has taken out this cysteine and replaced it with a serine, about the same size. So you don't have this disulfide bond. You also don't have that CX3C motif anymore. So one of those things is responsible for knocking this down. But this experiment indicates that this region is probably very important for binding and for infection of those human airway cells. So I don't show it here, but another virus from Tom Oomans, where he actually inserted an extra amino acid between the 2 cysteines, had a similar effect, poor infection in the HAE cells. So is CX3CR1 the receptor for RSV on these HAE cultures? There are several other possibilities, and exon 2, surfactant A have been proposed as a receptor, but Ralph Tripp had shown that G binds to CX3CR1, the receptor for the 1 fractalkine chemokine. And he also has shown that the G protein binds to lymphocytes and changes their migration patterns. 131.2G, which I've just shown you, neutralizes the virus in these human airway cells, and I just showed you it binds to the neck up in that conserved region. And Ralph had shown that it blocks the G protein interaction with CX3CR1. So we knew quite a bit about it at that point. And I've just shown you that the mutations there affect infection. So if CX3CR1 is the receptor on these HAE cultures, we should find it. And we should find it particularly on the ciliated cells, because those are the cells that are being infected. So this was our picture, one of our best pictures here in these cross-section of these cells. And what you see, at least if you're near the front, is that the cilia are the only things staining. These are kind of flopped over here. But in some cases, you can see that the staining is right at the top. So it is. The protein is in the right place at the right time to be the receptor for the virus. Now, I presented this, I think 4 years ago at this meeting in San Diego, and since then, 2 other groups have confirmed this. This is just the most beautiful picture, so I wanted to show you here. This is from Sanofi, Pasteur, and Genzyme, where virus is bound to the cilia, to these HAE cells, and the virus is stained here with a whatever color that is, green here. And the receptor here is in purple, so you can see some kind of purple color here, and tubulin is in the middle of the cilia. So all 3 are localized at the tip of that cilia. So it confirms very well what we had done also. So if CX3CR1 is a receptor on these cells, an antibody to CX3CR1 might inhibit, should inhibit, if it binds to the same site that the virus binds. So when we did the same experiment, but we put the antibody on the cells first, it had no effect on inhibiting infection of HeLa, but it did inhibit HAE cells. And we did expect to see more of an inhibition, but the problem I think here is that there's mucus above these cells, and it's difficult to get things through that mucus to get down to the cell surface. So CX3CR1 does look like a receptor for respiratory syncytial virus on these primary human differentiated epithelial cultures, and then because they look so much like in vivo, we expect it's also the receptor in vivo. So if it is, what about in mice? There is a knockout for CX3CR1. So here is the wild type infection of mice. Here's the knockout infection. It's a reduction of about 2/3 in the titer of virus produced in the lung. So it doesn't go down to 0. And we expect that this is some sort of a background level, perhaps the F protein, as Jose mentioned. Without the G, the virus is still infectious a bit. So we think that's probably what it is. It's also possible CX3CR1 in mice is different from humans, so maybe we wouldn't expect such a large reduction. But this was repeated 3 times and has a very strong probability. So our conclusions is CX3CR1 acts as a receptor. The neck of the protein is probably important in that binding. There may still, though, be other receptors. So I want to tell you quickly another story. I'm running out of time, but one of the things we had recognized was that when we grow the virus in vero cells, that virus does not infect these HAE cells very well. This is Jackie Corey's work, another grad student in the lab that just finished. So what happens is, here you can see from an earlier paper, virions grown in HEP2 cells have a 90 kilodalton G, but if they're grown in vero, it's 55 kilodaltons. We put a tag at the end of it so that if this is a cleavage, we wouldn't see staining of the shorter version. It would be cleaved somewhere here, losing this tag. And in fact, that's what happened. We could stain the large version, but the version that was in the vero cells did not stain. So there's a cleavage event here. Okay. So does cleavage affect function? Yes, it does. I had mentioned this before, but Here's the data. So if the virus is derived from HeLa cells, it infects well. If it's derived from Vero, it infects poorly. And this is generally 5 to 10 times difference. Yes, okay. So which amino acids are important for cleavage? We made a series of alanine scanning mutations and compared them to the wild type. So here's the wild type, grown in Vero. It's got a small amount of the large and a lot of the small. So when we scanned, we estimated it was somewhere around 210. We scanned up to this point, no difference in that cleavage pattern, but there were 2 mutations here which brought most of the protein back up, prevented cleavage. So this is 208 and 209. There are a couple others further down, not the next 2, but the next 3. So here's a summary of it. The bold ones are the ones that are important. We also identified cathepsin L as being the protease that cuts it. Now, cathepsin L cleaves at a hydrophobic followed by a polar, which would be exactly after those 2 most important amino acids. Now, when we fix this in the virus, we can then show the same experiment I just showed you. In HeLa cells, you grow it, it infects both. So the wild type and the mutant both infect well because it's not cleaved. In Vero, the G is cleaved, infects poorly, but when we fix that and prevent cleavage, now it infects much better. So what's the point here? The relevancy. Right now, if we make a live attenuated virus, grow it in Vero cells where it grows best, we have 1 dose. If we use a virus where this is fixed, we have 5 doses. 5 happy kids. So we think that's going to be important to build that into strains of the virus. And just the last slide here is just to show you, there's our cleavage. And we're wondering now if this C terminus has an effect, because in fact, if it's cleaving here, this whole structure that I spent the first half of my talk telling you about is still there. All we're missing is this part. And so it looks like this part of the protein is going to be important for something. Folding, interacting with this receptor CX3CR1, or something else. We're guessing, but we think that's important. And this is also the region that Jose mentioned where these 2 inserts have occurred in the Buenos Aires and in Ontario strains. And so that may actually be an important reason. I mean, it may be in the same region and causing problems there too. Okay, thank you.
Thank you. Wonderful talk. Have you been able to determine or check on non-ciliated bronchiolar epithelial cells to see if they express CX3?
No, we haven't. There was an early study that we did where we looked at the proteins before the cells before they had differentiated and had cilia, and they're very poorly infected at that point. But we haven't gone back and stained to see if they might be on the surface of those cells. I expect they wouldn't be.
This is a question concerning glycosylation of F and G glycoproteins. You said 60% of molecular weight or mass contributed by only carbohydrates. Has anybody looked at the specificity of antibodies to the glycans? As you probably know, in HIV, this is one of the hottest fields ever encountered, because antibodies which bind to carbohydrates may prevent the infection, may neutralize the virus, or may protect the virus from attachment of other antibodies. And with glycosylation that extensive, your protein core may be actually covered by carbohydrates, and antibodies don't even recognize that. Yeah. Has anyone looked at the importance of glycans in binding or neutralization to RSV?
I have not, but I think that's a great point. I expect that much of the carbohydrate-covered region is not going to be immunogenic, but it's possible. It's It's possible, yeah.
It's not on. Let's see. Does it matter where you get the epithelial cells from? Which part of the respiratory tree?
Yeah. It doesn't appear to. Ray Pickles looked at that early on from different sections. The one thing we have noticed, we don't have many children's lungs that we have isolated the virus from, the cells from, but in those that we do have, it looks like they have higher cilia and they're more highly sensitive to infection. It's kind of interesting.
Mark, lovely talk. Sorry, here, Ulster Power, Queen's University, Belfast.
Yeah, there you go.
Listen, Mark, we find when we use the differentiated cells, we don't get all of the ciliated cells infected. Do you see the same thing? And when you're looking for CX3CR1, do you see staining on all ciliated cells? And if yes, how do you reconcile the presence of CXCR1 on all ciliated cells but not getting all cells infected?
I don't know. And that's an interesting observation, but
Talk to you later.
we haven't looked at that carefully.
But again, do you see because we also have very little experience, but we have some experience with this type of, of cultures, and it's true that what Ulta was saying, that you don't get 100% of the cells infected, right?
Yeah. Well, yeah, and it depends on how much virus you put on. Depends on. We find it depends on the donor.
Yeah.
Donor to donor, some are more sensitive than others for an unknown reason. Maybe the receptor on the cilia.
Last question here.
So I see a lot of bronchiolitis and infection of those cells, and I'm still scratching my head on how those are getting infected. Club cells, any sort of bronchial or epithelium that's lacking cilia, that's I'm still trying to wonder these different cell subtypes that do get infected. I'm not sure if club cells do, but non-ciliated bronchial epithelials, resulting in the bronchiolitis is certainly a problem. I don't know. Do you have any thoughts?
No, well,
Okay.
it could be yet a different receptor, or it could be the same one on those cells. We haven't looked. These are difficult to make, but you can do it. Many of those other cells are difficult to grow, so most people haven't studied it. But if you can grow them, even for a short period of time, it could be tested.